In this video I want to show you how to find cells undergoing mitosis in an onion root cell. This is an allium is sometimes what it's called. It's just the tip of a root. Obviously that root is growing, so in order to grow it's got to undergo mitosis. Let me zoom out here. So that's the root. Every single one of those little pink dots you see is the DNA, which has been stained with kind of a purplish pinkish stain that specifically targets genetic material to help you see it better. And you can see the margins are, the edges are green. That's the cell walls. We're going to go ahead and zoom back out. We're going to go to yellow. Remember that's 10x now. We were on 4x. Right now 100 times total magnification, and then 40x or 400 times magnification. That's what you definitely need to be on in order to see these chromosomes. And then you also want to zoom in on your phone if you're using your phone, or you don't obviously can't zoom when you're just using your eye. We got a lot of good stuff going on in this slide right now. I'm right now on a cell undergoing metaphase. Hopefully I can find that one when I come back to it. I want to point first to this one right here where the pointer is on. That cell is in interphase. In fact, most of the cells on this screen right now are in interphase. If you look at the top left of the screen right now, most of those cells are in interphase. You can tell because the DNA material is diffuse, or it's spread out throughout the cell. It has not yet started undergoing prophase, which is when that nuclear envelope or nuclear membrane dissolves, and that DNA starts to condense into the sister chromatids, or into the chromosomes. So if you've watched that video, you know what I'm talking about with that. Right to the left, these two are mediocre to bad examples of prophase. Let's see if we can find a nice prophase. I'd say this one right here is a decent prophase. You can see that it's not evenly spread out anymore. You can notice some dark spots of the chromosomes. Maybe this one. I think this one's a nice one actually, right here. You can see some dark splotches and dots. Let's look around a bit more. Oh, here's a really nice one right here as well. So, right there, you can notice that it's, once again, it's not evenly spread out. That's because that DNA is starting to condense or clump into those chromosomes where it gets nice and wound up and organized. That's prophase. Metaphase is that one that I was on earlier. This is, oh, actually accidentally put it on to metaphase. That's a bad example of metaphase. It's not perfectly lined up. Let's see if we can find that metaphase from earlier. Oh, I think this is it right here. This is a really nice metaphase. It's kind of curved, but you can see that all the chromosomes have lined themselves up in the middle of the cell. This right here is another really nice metaphase that actually, it looks like anaphase is maybe just starting to happen. Oh, this is the one I'm on right now, is a really good example of prophase. The one the pointer's on, prophase, when the DNA is condensing again. Let's scan it on through here. And right here is another nice example of metaphase, all lined up in the middle. And then right below it, that's where anaphase is maybe just starting to happen. And over here, we're in the middle of anaphase. So you can see the chromosomes have now been separated. Up here, also metaphase, or sorry, anaphase, where the chromosomes are being pulled apart by those spindle fibers. Let's see if we can find any more nice anaphase. So one thing you're noticing, you just have to do a lot of scanning around. This will take a little bit of work. And I don't want you just to watch this video. I want you to kind of see this for yourself. Oh, look at this. Beautiful example of anaphase there. You can see those chromosomes are opposite each other, the ones that look kind of the same length, and they are being pulled apart. You can see that gap in between them. This one right here is maybe a kind of messed up. It looks like that one might be actually going wrong. Something's messed up in that cell. Yeah, that might be it for anaphase for now. Here's another anaphase going on. But once again, something might have gone wrong, or maybe we've got a weird shot of the weird cross section of this cell. Looks like those are separating. So now we want to find a telophase. Remember, telophase is at the end of anaphase. It's when the chromosomes have been separated and then they want to actually divide that cell. That's the process called cytokinesis. Just basically means cell being cut or cell being split in two. Oh, here's a nice little example of it. Right. Oh, just past it. Right there. The reason, actually, there's two of them in a row. I'm pointed at the middle of one, and then if you look above that, there's another one that just went under cytokinesis, or at the end of telophase. The way you can tell that it's not just an individual cell by itself yet that's in interphase is it's not a nice round nucleus. You can still see the chromosomes kind of lined up in a row, but right in between them you can see the cell wall reforming. So here's the cell wall reforming right there, and back down right there's another cell wall reforming. So those are almost totally done new cells. And on our way up here, I'll point out another really nice anaphase, unless that's the one I saw earlier, but it's too good to not point out. Let's see if we can find one or two more telophase examples. And this is probably easier with just looking through the eyepiece, because you can scan a little bit more of the field than what I'm looking at right now. I'm not finding any more telophase, or really obvious telophase, at least right now. Oh, look at that right there, the one that the pointer's on. Same thing, you can tell that it's not a nice circle yet, it has not reformed into a nucleus. These two right here have undergone anaphase and telophase, and they're just about done with cytokinesis. You can see a nice cell wall, nice cell wall reforming right there. So those are at the end of mitosis as well. I think I'm going to go ahead and stop it there. Hopefully that gives you a good idea of, in a plant cell, what you're looking for with mitosis. I'm going to really quickly put an animal cell on here to finish. I started with a plant cell because they're way easier to start with, and just bear with me for 1 second. I'm going to make this all one video. The plant cell, the animal cells one can be pretty hard to find. If you look at the slide itself, the dots are very, very small. I'm scanning around right now and you don't see anything. Let's see if I can locate it on here. There's some pretty small dots. If you look at the label on the slide, it's called blastula. That's actually the term for a clump of cells that are reproducing really quickly. So quite a few of these should be in mitosis. And was that it? No, that was not it. Okay, I'm going to pull the microscope slide off here for a second. Okay, they're kind of on the mid-right side. Okay, so if nothing else, it shows you that it can be kind of hard to find that. Hopefully you're not getting motion sickness watching this. Where is my blastula? Could be that I'm not quite in focus. Let's see if that's it. I'm kind of zigzagging, that's what I tend to do when I'm not sure where something is. Wow, okay. I might have to, oh, there we go. So, that was kind of tough to find, so hopefully you can take that as a lesson that you're not going to get this super quickly, and if you do, good for you. This is a blastula. It started out as one individual cell that was the union of a sperm and an egg cell, and it started rapidly dividing and dividing. That process is just called mitosis. So compared to this is growth, just like with the plant cell, except for this is growth kind of from one individual cell, and the plant cell was the root growing to extend further into the ground. So we'll go to yellow. I just zoomed out too, that's why it doesn't look like we're any closer. And we'll go to blue. And then I'll definitely have to, you notice you really can't see much yet even, so we're going to go ahead and zoom in even more. And I like to start with the plant cell because I think it's way easier to see what's going on in the plant cell than in these animal cells. Right now, this cell and all the ones around it are in interphase. You can't really see the DNA because it's being evenly spread out. Right here, notice it's considerably, I think it's kind of smaller than the plant cell in my opinion, but this right here looks to be in metaphase. They're all those chromosomes are lined up in the middle. Let's look around. Here's another nice, this is probably anaphase, I believe, two sets of chromosomes being pulled apart. Over right here looks like another nice metaphase. And what you're seeing is that it's a lot harder to kind of see what's going on here with these animal cells. Another nice metaphase right there. Oh, look at that beautiful metaphase, perfectly lined up down the middle there, and you can actually see those spindle fibers really well. They're going to start pulling the chromosomes apart. It's just not as much to see on these animal cells. Once again, this is a fish, the fish blastula. I'm really having trouble seeing much of anything beyond metaphase. Maybe anaphase is just starting there. Yeah, it's just not a ton to see. But I do want you to take a look at this one as well. Okay, so that's it for now. Sorry it was such a long and dithering video.